Dear students, in this video we are going to discuss about the calculation of total RBC count. So this is a very important topic in hematology. So I will discuss in the easiest form how to calculate the RBC. So we know that RBC counting is done in the Newber's chamber. Total count of RBC is done in the Newber's chamber. This is the picture of a Newber's chamber. And if we look into this, we know that in the central part of the upper surface of the Newber's chamber, there are two central platforms. We can see here two central platforms and two lateral platforms. Okay, two central platforms we can see here and two lateral platforms, and they are separated by actually the gutters or trenches in the form of H. So we have two central platform, two lateral platforms. In the picture below we can see also the central platform and two lateral platform, and they are separated by trenches or gutters, and it looks like an H. Okay, now the transverse gutter in the middle part of the H, it separates the two central platform, which actually contains the counting grids. Okay, so the central portion of the H is separating two central platforms, and these central platforms they contains mainly the counting grids. Okay, now the two central platforms are separated from the lateral platforms. The groove in between them, the trenches, it is known as the moat. Okay, so and when a cover slip is placed over this central platform, you can see in the picture below, when a cover slip is placed over the central platform, the lateral edges of the cover slips, they are supported by the lateral platform. So lateral edges, they are on the lateral platforms. Okay, but the central platform you can see here, it is not at the same level of the lateral platforms. It is a bit depressed. How much depressed? By 1/10 millimeter from the lateral platforms, so that the undersurface of the cover slip lying above the central platform, it is exactly 1/10 millimeter above the upper surface of the central platform. Okay, so there is a gap or depth of 1/10 millimeter, that is the depth is 0.1 millimeter. Okay, so to summarize, we have two central platforms which has the counting grids. We have the two lateral platforms, they are separated by the moats. Okay, and when we are placing the cover slip, the lateral portion it is supported by the lateral platforms, whereas the central platform, it is a bit depressed. How much depressed? It is 1/10 millimeter, or the depth is 0.1 millimeter. So this is, we know, we have already seen. And when we will form the solution with the RBC fluid, so we will charge it into in this central portion. Okay, if the charging it goes into this surrounding trench or the moats, then it will be called as the overflow. It should be exactly on the counting grid, the charging. Now, if we focus this counting grid under microscope, it will look like this. Okay, so it is the thing that we will look under the microscope if it is focused under the low power. Okay, in the low power objective, it is focused, and we can see this counting portion. So if we look into the counting portion, you can see that it is a bit like square like structure, which are the length of the square is 3 millimeter and the width of the square is 3 millimeter. So the total area of this counting grid is 3 into 3, that is equal to 9 millimeter square. And this 9 millimeter square is divided into 9 boxes. Okay, so 3 boxes horizontally and 3 boxes vertically. So we can say that each of these box, they are of 1 millimeter. So 3 is divided into horizontally 1 millimeter, 1 millimeter, 1 millimeter, and again 3 in the vertical 1 millimeter, 1 millimeter, 1 millimeter. So each of these large box, they contain, they are length is 1 millimeter and breadth is also 1 millimeter. And we can see here this 1 millimeter distribution. Among these boxes, the 4 corner boxes, they are used for the calculation of WBC. Whereas the central 1 millimeter square area, it is used for the counting of RBC. So here I want to mention one important point. If I want to count WBC, I have to see the whole 9 millimeter square area, the larger area. So I will focus under the low power microscope. Whereas if I want to calculate the RBC, I want to see the focus on this RBC counter. So I have to focus on a narrow area of 1 millimeter square. So for that reason, for the counting of RBC, you have to focus into the high power objective. So it's clear. So for counting the WBC, you have to focus on the low power objective. For counting RBC, you have to focus on the high power objective 40x. Okay. Now if we focus it under the high power objective, and we will see that this 1 millimeter square area, the RBC counting chamber, it is further subdivided into 25 boxes, large boxes. 25 boxes means 1 millimeter is divided into 5 parts. So each of these large boxes, they are of 1 by 5 in length and 1 by 5 in breadth, because 1 millimeter is divided into 5. So 1 by 5, 1 by 5, 1 by 5, 1 by 5, 1 by 5. So each large box, they have a length of 1 by 5 by 5 millimeter and breadth of 1 by 5 millimeter. Among these 25 boxes, we count the WBC into RBC into 5 chambers, that is upper left, upper right, lower left, lower right, and the central. Okay, we can identify the upper right and upper left. How can you identify? In the upper left, there will be no boxes in the above and to the left. In the upper right, there will be parallel lines and no boxes in the upper, above, and to the right. Okay. In the lower left, there will be no boxes to the left and below. And the lower right, there will be no boxes. The parallel lines will be there in the lower part and to the right. And the central chamber will have 2 counters each on above and below. So in this way, we can identify which one is upper left, upper right, lower left, lower right. Okay. If there is no boxes up above and to the left, then it is upper left. So now we consider the upper left. So we know the area of upper left is 1 by 5 into 1 by 5. That is equal to 1 by 25 millimeter square. Okay. And we already know that the depth is already, it is made in such a way, the depth is 0.1 millimeter or 1 by 10. So the volume of this upper left, we can, or a one large square, we can say that it is 1 by 25 millimeter square into 1 by 10 millimeter. That is, the volume of one large box will be 1 by 250 millimeter cube. Now, how many boxes we have taken? We have taken upper left, upper right, lower right, and the central. So 5 boxes. So the volume of one large box is 1 by 250 millimeter cube. So volume of 5 large boxes will be 1 by 250 into 5. That is 1 by 50 millimeter cube. Now, suppose you have counted. Now you have done your counting. You got 150 cells in the upper left, 120 cells in the upper right, 130 cells in the lower left, and 140 cells in the lower right, and in the central it is 110. You have to add all these numbers of cells. So we are getting 650. This is the total number of cells which are distributed into these 5 large boxes. Okay. So we can say that we know the volume of 5 large box, that is 1 by 50 millimeter cube. Okay. So 1 by 50 millimeter cube, that contains 650 cells. So 1 millimeter cube will contain 650 divided by 1 by 50 into the dilution factor. We have to multiply it with the dilution factor to get the total count of RBC. Okay, so dilution factor in case of RBC, it is 200. How can we get it? We get it from the RBC pipette. If you see, look at the markings of the RBC pipette, it is 0.5, 1 and 101. And this marking, to be noted that these markings 0.5, 1 and 101, they have no standard units. They don't have any units like millimeter cube like this. They just indicate the relative volumes. Okay, so you have to say what is the marking? It is 0.5 unit, 1 unit and 101 unit. Okay, now we know that the blood is drawn is up to 0.5 and marking, and the RBC fluid is drawn up to 101. Okay, so and we usually discard initial few drops. So in the stem portion there will be no mixing of fluid with the RBC. So what to have an accurate counting, because the initial part of the fluid may not have the RBCs, may not contain any RBC. That is the reason we will not take the part of the fluid up to 1. Okay, so we will say that 0.5 unit of blood, it is diluted into 101 minus 1, that is 100 units of fluid. Okay, so 1 unit of blood will be dilated by in 100 divided by 0.5, that is 200. So this one is your dilution factor. Okay, we know that now. So we already counted, we have 650 number of cells, and that is distributed into 1 by 50 millimeter cube. So 1 by 50 millimeter cube contains 650 cells, and 1 millimeter cube contains 650 divided by 150. That means 650 into 50 into the dilution factor, that is 200. So 50 into 200, that is 10,000. But in your examination, don't go directly to this number of 10,000, because examiner will always ask you from where did you get that 10,000 amount. Okay, so in that case you have to explain it in this way. So the total count of RBC in this case is 6.5 million RBC per cubic millimeter. And always remember, when you are telling the RBC count, always mention the unit. Okay, thank you.